three former graduate students here besides Ray and Linda. Linda is retired now from Western Kentucky. I'm still at the University of Georgia, and I'm kind of beginning to think maybe it's my time to check out the University of Georgia as well. But Chris Agee with Pennington Seed Company also is one of my former graduate students. I served on Linda's committee and Ray's committee, but was a major professor for Chris Agee. You're getting a handout. I brought 60 handouts, and if you're registered for the meeting, I think there's 57 of you. You're welcome to take one of these. If you're not registered, please let the registered people get one. And if there are any left over, we'll try and share some. And if you want some, we'll give them to you, or we'll get one to you. But this is a company I started in 2001. And so we've been in business now for 17 years. And the technology that I'm going to tell you about today is something that's actually an international standard. So we went through the process of comparing it to other systems that we have for analysis and make sure that it was at least comparable and in some cases better than the existing technology at the time. So I'm going to talk to you today about seed quality and the testing that gets done on these novel endophyte products to provide assurances that when you buy this seed at a value-added price, that you're actually getting what you expect to get when you purchase that seed. Okay. So here's an example of a tall fescue field that's in not very good condition. That particular field was planted to endophyte-free tall fescue back in the late 1980s. This picture was taken in the early 1990s, and you can see it's very thin stand. And actually what you see there, those remnant plants from the endophyte-free field are actually infected with toxic endophytes that had regrown from seed that was in the soil. And if you look at the old Kentucky 31, at one time we were recommending go out and plant endophyte-free tall fescue seed, so get some Kentucky 31 that's endophyte-free and plant it. Well, we found in not too long a time that what would happen is that endophyte-free tall fescue would die and the endophyte-infected wild types persisted. So we started asking questions about what's the value of the endophyte at that particular time. This is in the late 1980s, and we were able to demonstrate that having endophyte in there provided various competitive advantages compared to the endophyte-free tall fescue, particularly in drought conditions. And we all know that periodically we're going to get some pretty severe droughts, and we need to utilize our forage out there during those drought periods. And if you've got endophyte-free and you've got the drought stress plus the animal stress on top of that, then you're going to take out your tall fescue. Having the endophyte in there provides you some advantages to resistance against that event. So, what is a normal seed test? And I've got a picture of a label off of corn. And you're sitting there saying, well, we're talking about tall fescue today. Why are you telling us about corn? Well, the seed requirements, these are federal seed laws that are on labels for corn, are the exact same seed laws that are on labels for tall fescue. So that means that there's no seed laws requiring testing for endophyte or providing some information regarding the amount of novel endophyte that you might find in your tall fescue seed. So some of the things that federal seed laws ask you to identify are the species, the variety, the lot number, the date that germination purity. Noxious weeds is one that you really want to pay attention to. Those are difficult to control weeds. And if you've got difficult control weeds in your seed bag, then you're planting difficult to control problems out in your pasture. Inert matter and all this. The origin, the date of testing has got to be on there. Any seed treatments that you might have. Okay, so this is federal law. The only laws that are out there now that require endophyte testing is that if you're making claims to endophyte-free seed, which is a carryover from the 1980s, there are some states in the United States that require you to prove that the seed is endophyte-free, and anything over 5% infected is considered endophyte infected, okay? And that would not sell as endophyte-free seed. But we don't care about endophyte-free seed because it's not going to persist in the pasture. We're talking about novel endophytes that have got the toxin removed and the advantage of having the endophyte in the seed. So there's no regulation for labeling seed for novel endophyte technology. So the endophyte, which Craig has already talked about, this is a picture that one of my graduate students took way back when, and the endophyte is actually growing in between plant cells, and any nutrients that are leaking in between the plant cells is what the endophyte utilizes to, for its sustenance. Okay, there's another picture that we took a long time ago that you've seen a number of times in the presentations today. This is a different type of staining technique. Okay, and just to remind you what the life cycle is, if you have an endophyte in a tall fescue seed and you plant it and the endophyte is viable, it's going to concentrate down around the base of the tall fescue plant. And then as the plant grows a seed head, it's going to move up into this seed head, and then it's going to transmit to the seed, and then you plant that seed and you get endophyte infected tall fescue again. So the point here is that the seed is the transmission vector here. This is the way it gets dispersed, okay? Another important thing to consider is that this endophyte is totally dependent upon the grass plant for its sustenance, and it's developed this nice symbiotic relationship with the tall fescue plant. So it is not to its advantage to go through a sexual replication, okay? It doesn't go through a sexual exchange, because if it does, it's changing, and then it might not be a good host, or the endophyte might not make a good marriage with the tall fescue plant. So it's what we call conserved genetics, in that it's going to stay the same from generation to generation to generation. And then finally, whoops, that means that if you've got a toxic plant with a toxic endophyte in it, and you harvest that seed, you're disseminating toxic tall fescue seed and toxic endophyte. But if you have non-toxic, you're producing non-toxic seed and you're disseminating non-toxic seed as a result. Okay, so what we know is that 95% of the Kentucky 31 tall fescue pastures that we have out there are infected with these toxic endophytes. Okay, and these endophytes, Craig showed that chicken wire graphic earlier about what the toxic endophyte, or what the toxins are. These are chemical derivatives of LSD, the hallucinogen. So your animals are consuming these toxins that are LSD-like compounds, okay, and that's what's causing the abnormality in your animals. But the novel endophytes don't produce these toxins, so consequently you get the benefit of the endophyte without the toxicity. So Craig showed this slide earlier also. We can take toxic endophyte, we can remove it, and then insert non-toxic endophytes or novel endophytes in there, and therefore we have these approved varieties that are, or improved varieties that are novel endophyte infected tall fescue. Novel endophyte infected tall fescue, or just because you have an endophyte that's not toxic, does not mean that that's a good candidate for putting into pasture plants. Not all endophyte plant associations are equal. Okay, so the plant breeding companies that have got their products here today, they've gone through the process of finding good endophytes that will transmit, that do have a happy home in the improved varieties. So it's a specific interaction between the endophyte and the tall fescue plant that results in this product that you're going to, or products that you're going to hear about later on. But even then, even then, harvest conditions, if they go out and they harvest the seed too early and the seed is still green and they bring it in, the endophyte transmission is the same going into the seed, but the viability in the seed changes. So the endophyte may not be as viable in the seed, and it certainly doesn't last as long during storage conditions if they don't harvest the seed at the right stage. Okay, and then storage conditions are also important. If they store at high temperature or high humidity, chances are that the endophyte viability is going to be compromised in that seed. So they have decided to do some quality testing. This is what our test looks like. It's kind of the same concept as home pregnancy testing. Okay, this is immunology-based testing. So here's a membrane here, or a image of endophyte-infected tall fescue seed. Here's an image of endophyte-free tall fescue seed. That particular lot right there was 91% infected. This is endophyte-free. Not only that, but we need to start asking the question, how many of these endophytes that are in the seed are actually viable and being transmitted to the next generation of plants? So we also have a test for that, which these companies do. And these little circles that you see here are from plants that we've grown in the greenhouse. And at the stem base, we've made a horizontal cut. And we just get a little wafer of that and place that on this special tissue that we've got here, a special membrane. So these are basically just circles or cross sections of stem bases, okay? And anytime you see pink like this, that means the endophyte is present. And down here you can see that there's no pink color. There's some discoloration there, but there's no pink color. Those are endophyte free. Okay, so we know there's endophyte in the seed. We know that there's endophyte present in the plants that we're growing in the greenhouse or we're testing for viability. What about toxicity? Okay, so this is an image where we test for toxins. And you can see that there's 12, these are little reaction vessels that are in a plastic plate about this big. Okay, and each one of those spots right there, we can test a seed or we can test a plant for toxicity. Okay, and if we get a yellow color on our test that we developed, that means it's non-toxic. And if we get a clear reaction vessel like that, that means it's toxic. So this up here are all non-toxic, and this came from, this is actually seed that we tested, came from seed lot that was 92% infected. All of those infected seeds were toxic, okay? Now, if you go out and test your pastures, you're probably, and if you've got Kentucky 31, you're probably going to test somewhere between 1,000 parts per billion to maybe 10,000 parts per billion, or 10 parts per million is what we've seen. Okay, this test right here turns clear like that. Remember, this is non-toxic. That turns clear like that at 25 parts per billion. So we're about 1,000 times more sensitive in terms of testing for toxicity than what's going to be present out in your pastures or out in the seed. So when we say that this is non-toxic, we know that it's non-toxic, okay? So in addition to getting all of this stuff on your seed test, okay, if you buy seed that is Alliance approved, you'll see this image right here, and Jimmy was talking about that. And if you read, it says, this seed lot has been tested and determined to contain at least 70% novel, meaning improved endophyte, and no more than 5% off-type endophyte or toxic endophyte. We've been testing for the Alliance now since they started. I don't think we've had one seed lot yet that has gone commercial that's tested more than about 0.5% off-types. They do a really good job of keeping their fields clean and non-toxic. Okay, so you want to be looking for that label right there to assure that the quality is, to make sure that the quality is there that the Alliance has established. So just look for that logo when you go shopping for seed. And with that, ooh, it's lunchtime if there are no questions. I think they're hungry.